Alrighty, so like I said, there is a quiz on Friday. Today we are talking about active transport. So, since last week, we've started talking about membrane composition, we've talked about passive transport, and now we're going to talk about active transport. Those three things are what is going to be covered on your quiz on Friday. We will review for said quiz. Today is Wednesday, tomorrow. So, today, my goal is for you to be able to differentiate or tell the differences between active and passive transport. So, does anyone have any questions? Alrighty. So, a little bit of review. This is what we've been doing our whole lab over, everything that we've talked about since we finished up membrane composition. Passive transport is the movement of materials across the cell membrane from an area of high concentration to an area of low concentration. I should say from. That's an important distinction. From an area of high concentration to an area of low concentration without using energy. This is important. We got to know where our ions are moving. And they are always going to move from an area of high concentration to an area of low concentration. Yes. Okay, this is review. So if you've got this, you don't have to write it down. But if you want to make sure this sticks, I'd write it down. Thanks for asking, please. All right. So passive transport is the movement of stuff from an area of high concentration to an area of low concentration. So let me see, draw a little picture. I guess I could use the pen so it translates via everything else. All right. So let's say we've got a little cell membrane. Yes, please. Let's say we've got a little cell membrane. Goodness gracious. This is outside the cell. This is inside the cell. We've got a lot of stuff inside the cell and only a few things outside of the cell. If that is able to freely move, will they move in or out of the cell? I think I heard it. Out. Movement is going to be out because they're going to move from an area of high concentration to an area of low concentration. This does not take energy. Now, active transport does take energy because active transport is moving materials against this concentration difference. Moving materials from an area of low concentration to an area of high concentration, like herding cattle into a small area. Because we want, sometimes that's what the cell needs. Bless you. Sometimes the cells need to move things against their concentration gradient. This is important. Make sure you get this down. So, if we have our little cell here, this is outside the cell, and this is inside the cell. And we've got a lot of, let's say, sugar on the inside, and not a lot of sugar on the outside. Sometimes our cells just need sugar, right? Sometimes they don't have enough of it in order to get energy and do the cell things. So they will move things from an area of low concentration to an area of high concentration in the opposite direction of diffusion, working against diffusion. This takes energy. If you had 100 kindergartners and you put them in the cafeteria and nobody was around, those kindergartners would probably spread out throughout the whole school, right? That's diffusion. That doesn't take any energy. It doesn't take any energy to watch kindergartners run around. But if we wanted to take them from wherever they are in the school and put them into the cafeteria, That would take some energy. That would take some work. That is like active transport. It is moving things against that concentration difference. So moving things from an area of low concentration to an area of high concentration. And we call it active transport. Active transport. Questions here. This is kind of the opposite of what we've been talking about all week. Active transport is moving things from an area of low concentration to an area of high concentration. This takes effort. If we released a bunch of crickets in this room, they would naturally just kind of spread out and take up the whole space of the room, right? But if we got annoyed by all those crickets and wanted to track them all down and put them back into their box, that would take a lot of energy. The cells do the same thing. So, this requires energy. Can I get the cell phone put away, please? Thank you. This is new. There's a quiz on Friday. Respect your time, respect my time. All right. 2 types of active transport. There are 2 big categories of active transport. Just like with passive transport, we had diffusion and facilitated diffusion. With active transport, we have molecular transport and bulk transport. Both of these are types of active transport. I want to talk about them both. So, these are our 2 big categories. And I will help y'all organize these in your head at the end of the class. So, if you don't get these, they will be the titles for the next 2 slides. There are 2 big types of active transport because some things take more energy than others, right? If we let a bunch of high schoolers loose in the building, it would probably be a lot easier to get them where they need to go than it would be a bunch of kindergartners. Some things take more work than others. So we have different types of active transport. The first is molecular transport. A molecular transport have special proteins that carry them across the membrane. They act like little pumps. Many cells use protein pumps to move calcium, potassium, and sodium, those little ions that can't freely move across the membrane. I don't know why there's a little, there we go. So, this right here, we've got let's say this is inside the cell, and this is outside the cell. And we've got our membrane here. And we've got our special protein. And that protein wants to move these out. Naturally, this would not be the way things work. So we are going to move them against their concentration gradient. In order to do this, this takes energy. This pump has to use energy to get these across that membrane. And they're going to do it one at a time. It's not going to be a lot. That is why we call it molecular transport. Yeah, small molecules, small things that are carried by protein pumps. Because they're sitting and they are pumping out the things that need to be pumped out and pumping in the things that need to be pumped in. Doing okay so far? All right. So this is molecular transport. This takes energy. These pumps are all sorts of throughout these cell membranes. Questions here? All right. So the other one is bulk transport. And here's what you need, right? You need this first sentence and just this first line, because we are going to talk about all those different forms. So bulk transport, that is moving a lot of stuff out all at once. If we had a really, really, really big molecule, it would not be able to fit through one of those small little protein pumps. But we still need big molecules in and out of our cells. We still need those big molecules in and out of our cells. So we've got a different way to get them where they need to go. And we call this bulk transport. We are transporting things in bulk. It can take a lot of different forms. We're going to talk about 3 of them specifically. And this all depends on the shape and size of what we are moving, as well as if it is moving in or out of the cell. Because it's going to look a little bit different if we're moving stuff in versus moving stuff out. So this is the big stuff. Hence bulk transport. All right. Bulk transport. We're going to talk about these different types of bulk transport. The first of them being endocytosis. Endocytosis is the process of taking material into the cell and making a little pocket on the cell membrane. What's going to happen is the pocket will break loose from the outer portion of the cell membrane and form a vacuole or vesicle. So I've got a diagram on the other side. But just imagine, let's say this tissue is our cell membrane. Let me get eyes up here. This tissue is our cell membrane. Imagine it's a big old circle around here. And my clicker is what wants to come into the cell. Endocytosis, taking things into the cell. What's going to happen is that membrane is going to form a pocket around that little molecule coming in. It's going to close up around it. It's going to break off. And this is going to be, well, hopefully be intact. This is going to be our vesicle that's going to go around and take this to wherever it needs to be in the cell. And this will continue being a part of our cell membrane. Does that make sense? That is endocytosis. We form a little pocket. And once our pocket is full, it will kind of close in on itself, pinch off, and then be a little pocket or be a little vesicle or vacuole in the cell membrane. That is endocytosis. Endocytosis takes stuff from the outside and brings it inside. Endocytosis. And take a little pocket, and we are going to form a vesicle or vacuole and have that float loose in the cytoplasm. Take it where it needs to go. Endocytosis. Endocytosis. I say it a lot because these words are going to sound really, really similar, and saying it a lot kind of helps make that differentiation. So, endocytosis is the type of bulk transport. It is taking stuff in. There's a special type of endocytosis that we call, never mind, here's a picture. So this is exactly what I had described in my tissue example. We've got all of these little things that we want inside the cell. That cell membrane is going to make a little pocket. When that cell membrane makes a little pocket, eventually it will close in around it. When it closes in around it, that vesicle will be released and that plasma membrane will be still left intact. This is endocytosis. We're going to take stuff from the outside, fold it in, package it up, and send it on its way. There's a special type of endocytosis called phagocytosis. Phagocytosis is specifically taking stuff in and putting it in a food vacuole. It's a type of endocytosis. We've got an extension of the cytoplasm and the cell membrane that are going to surround a particle and package it within a vacuole, a food vacuole. The cell will engulf it, and then it will probably send it somewhere to be digested. This is how amoebas eat. A lot of single-celled organisms eat this way. And this is how blood cells destroy damaged cells. So our white blood cells are kind of our cleanup crew of our blood. And they have a special ability to take in and destroy damaged blood cells. And the way they take in these damaged blood cells is called phagocytosis. It is a special subtype of endocytosis. Doing okay so far? A lot of long words that sound the same. A lot of long words that sound the same. That is phagocytosis. Phagocytosis. Sometimes you will hear it pronounced phagocytosis. I think that sounds weird, and I was taught phagocytosis, so I say phagocytosis. Some people say phagocytosis. They're the same thing. Apricot. Potato. So that's phagocytosis. It is a type of endocytosis. I know the slides are long. It's a shorter PowerPoint. And it's a really big font. So that is phagocytosis. So if we got a little amoeba floating around in a pool of something, and it has located a molecule it wants to take in, it will perform this process. Again, phagocytosis, we've got our really, really large particle. That particle will hit the plasma membrane. The plasma membrane will fold around it, and then that is going to be sent to where it needs to go. So, special type of endocytosis. It's one large molecule, and it's specifically food. So, this can happen in the reverse direction, and it is called exocytosis. Think exhale. Exocytosis. I do the arm movements because it helps me remember. Exocytosis. This is when we release a material. We're spitting out stuff. During exocytosis, a vacuole will fuse to the membrane. That membrane will open and it will release whatever was inside that vesicle. It is the exact opposite of endocytosis. The diagrams look the exact same, just in an opposite direction. So a lot of cells do exocytosis, specifically cells that are meant to do some sort of secretion. So our pancreas, the cells in our pancreas make insulin that is released into our bloodstream to help balance our blood sugar. Those cells in our pancreas perform this process, exocytosis, in order to release that insulin. The process of those cells releasing that insulin is called exocytosis. Same thing with the cells in our skin and on our scalps, right? They secrete oils to keep our skin protected and our hair protected. Those cells in our scalps that secrete that oil do so by exocytosis. I know it seems to be a battle a lot of the time. It's exocytosis. This takes energy. All of these things, moving cell membranes around, getting vacuoles to where they need to go, all of this takes energy. So, this is our picture. This is our drawing. Did I go too fast? Just tell me. I'm more than happy to go back. I get ahead of myself. I get excited. So, this is the process of exocytosis. We've got our vesicle. It's going to fuse with our cell membrane. It's going to release stuff into wherever it's going to go, into the outside, into the out. Questions here? All right. We're going to make a concept map. And I've got them all printed out so you don't have to make the actual bubbles yourself. But we are going to map out our process of all of the cell transport we have done. Because we've talked about a lot of cell transport in a relatively short amount of time. And I think it's nice to be able to see this stuff written out. That, and I love a good concept map. So we're going to fill these out together, and you'll glue them into your notes. So, this whole thing, all of the stuff we've talked about since we started passive transport a while ago, all boils down to cell transport. Cell transport. It all boils down to cell transport. And there are two types of cell transport. What are they? Active and passive. So we have passive transport on the first one. And active transport. Active and passive. So all cell transport falls within these two categories. So we talked about active transport today. What were our two types of active transport? Molecular and bulk. So we've got molecular transport. My boxes are a little bit smaller than yours. And bulk transport. If you think it would help to leave a little space and write the definitions in here after we are finished, because we'll probably have some time, that'd be great. You don't have to, though. I'm not turning these in. These are for you. Okay, coming off of bulk transport, there were two main types. What are our two main types? Go start with the same letter. Yeah. I don't know. Okay, phagocytosis is a type of endocytosis, but we do have endocytosis. What was the other one? Yeah, endocytosis and exocytosis. And we already mentioned our specific example. We've got our example of endocytosis, which is phagocytosis. Here's phagocytosis. Can you read those? All right. Now we're going to time travel back in our brains until back to last week when we talked about passive transport. There was two types of passive transport. Do we remember those? Diffusion and facilitated diffusion. We went over it in our lab review yesterday. So we've got diffusion. Remember, that one goes straight through the membrane. And we've got facilitated diffusion. What's the difference between diffusion and facilitated diffusion? Campbell? Difference between diffusion and facilitated diffusion? Facilitated diffusion has to go through a special channel, or we use proteins in facilitated diffusion. And there was one big example of facilitated diffusion that we spent a very long time and a very stinky time talking about. What was that? Osmosis. So we have these concept maps. We can now use them to help us figure out, whoopsies, went back. We can use them to help us figure out what type of transport falls into which categories, right? So if you're thinking about moving large amounts of things, right, moving large stuff around the membrane, you're like, oh, okay, I know that's bulk transport. So you can go back and you can say, okay, it's active. That means it needs energy. And then you can tell whether or not it's going in or out, and you've got exo or endo. And if it's going in, it might be phagocytosis. It might just be generalized endocytosis. So this kind of helps organize what all we've been talking about the last couple weeks. Questions here? All right. There is an exit ticket on Canvas, nothing too crazy, couple questions. I'm going to pass out glue sticks for you to get these in your notes. And yeah, that'll finish us up for today. Yeah, you can put it in your folder if you want.